Okay, so stratified squamous epithelium. So stratified, lots of layers, squamous, squished, flattened cells. So down here, this would be our basement membrane, okay, down here. Up here would be our free apical space, okay? And between that, these are multiple layers. Okay? So stratified squamous epithelium, lots of layers of flattened squished cells. And it's actually more towards the apical surface is where you see that really squished, flattened appearance. Stratified squamous epithelium, where do you find it? Ideal locations, the epidermis. So when you touch your skin, you're touching your epidermis. Vaginal canal, the esophagus, on the tongue or lining the oral cavity, or the anal cavity. So anything that's open to the outside and subject to abrasion. So your epidermis, you scratch your skin, you put on and take off clothes, you dry yourself with towels. Anal cavity, feces, hardened feces have to pass through there. Vaginal canal for intercourse. So these are all areas that are open to the outside and subject to abrasion, so lots of friction. Well, it doesn't make sense if something is open to the outside and subject to rubbing forces or abrasion, friction, to have a epithelium like simple squamous epithelium, a single layer of cells, right? We would be leaking everywhere, or as soon as you scratched yourself, you would take off the only layer of cells that you had. So anything that's open to the outside and subject to abrasion is where you find stratified squamous epithelium. And so it makes sense because here, this would be our basement membrane, free apical space up here, lots of layers of flattened squished cells. And so whenever you have abrasive forces, cells towards the top, towards the free apical surface, can slough off. These are old dead cells. They can slough off. And then we have more that can be pushed up towards the apical surface and regenerate, okay? So when we lose some of these cells to abrasive forces, we still have some below it. And the reason why the cells that are towards the basement membrane maybe look more cuboidal-ish in shape, these are actually the stem cells that are dividing to form more of these cells, so that as these cells slough off from the apical surface, right, we still have some below, but we don't have limited layers. We regenerate more from these cells and they get pushed up, and then we regenerate more and more and more, okay? So but really it's the cells that are closer towards the apical surface that really give that flattened, squished appearance. So this is stratified squamous epithelium. I wanted to show you this image. This is also stratified squamous epithelium. Down here, this would be the basement membrane where the cells kind of somewhat look more cuboidal because they're generating more cells to be pushed up. And then as we go up towards the apical surface, you really start to see the cells that are squished, that irregularly shaped structure, okay? This up here where you don't see any nuclei, so these darker space, this is, you can see the nuclei in the cells of these layers, but up here you can't see nuclei. So it's because these are all dead cells. So this is an example of stratified squamous epithelium where it's keratinized. We'll talk about the process of keratinization in the integumentary system. But if you've ever had a callus on your hands, well, where do you get it? Why do you get a callus? Is because that particular area of your hand is subject to lots and lots of abrasive forces, more so than other areas of your skin. Well, that callus is this keratinized portion where you have lots of layers of dead, hardened cells for more protection. So this is it, but it's still stratified squamous epithelium, lots of layers of squished, flattened cells. So if I put the pointer here and ask what's the specific, you would say stratified squamous, and if I ask, and the general would be epithelium. Same thing for this. If I put the pointer here and ask for the specific, you tell me stratified squamous, and the general would be epithelium. Okay, it's just this stratified squamous epithelium lacks that big dead corneum or that keratinized layer. Okay, so where you don't see a thick keratinized tissue, like here, this would most likely be of the vaginal canal. Okay, this would be like the soles of your foot or the palm of your hand. okay, where it's subject to lots and lots of abrasive forces consistently. Simple cuboidal epithelium, so a single layer of cuboidal shaped cells. Ideal place to find simple cuboidal is in the kidney tubules, okay? So you want to look for a kidney slide. So here, this is a kidney tubule, there's just lots and lots and lots of them. Each one of these tubules on the outside would be the basement membrane, and on the inside right here, where you see this hollow space, this would be the lumen, okay, where the secretions or something is flowing through. This would be the apical space, right? It's open to the outside, right? So, and if you look, they look like little cubes, and the nucleus is very, very circular, and it's smack dab in the center of those cells. So lining each one of these tubes would be the simple cuboidal epithelium. So this is higher magnification, and even though these aren't nice and super spherical tubules, they're somewhat, elongated, but you can still tell, like on the outside, this would be where the basement membrane is, and then here, this would be the tubule, the lumen of the tubule. This would be that apical space. And look at those beautiful, simple cuboidal epithelial cells with that very spherical nucleus located smack dab in the center, okay? So if I'm asking cuboidal, it will be in a tubule shape because you are beginners, and that's how the easiest way to identify simple cuboidal epithelium. Let's talk about the functions. Anywhere where you have it mainly for secretion, so lots of secretion, it will be going on in simple cuboidal cells, but also some absorption. That's why you find the kidneys just full of simple cuboidal tubules, because the kidneys are filtering your blood. So things that need to be, the kidney tubules are going to be secreting things that we don't want, reabsorbing things from our blood that we do want. If you look at glands, so exocrine glands, things that secrete substances to the outside, sweat glands, sebaceous glands that produce oil to moisten our skin and hair, those exocrine glands you will find that will look exactly like this. You'll see little tubules and simple cuboidal cells lining those tubules because they're secreting a substance into this lumen to be directed to the outside. Now, endocrine glands like the thymus or the pancreas, you'll also find cuboidal type cells, but they won't be in this nice tubule form. So that's why as beginners, if I'm asking simple cuboidal, it will be in this nice tubule shape, okay? So you find it in exocrine glands and kidney tubules where you have lots of secretion going on, okay, and some absorption. It's because these cells are producing a product that gets secreted, and so they're a little bit fluffier because lots of production is going on in these cells, okay? And that product is being secreted into the lumen. So if I have the pointer here and I'm pointing to this layer of cells, the specific would be simple cuboidal and the general would be epithelium. So simple cuboidal epithelium, a single layer of cube-like cells. And I'll ask it in a tubule form. Simple columnar epithelium. So a single layer of tall column-shaped cells. Single layer of column-shaped cells. Here, this would be the basement membrane. And look, all of this below, this would all be connective tissue below. These would be a blood vessel below. You can see the red blood cells. And so nutrients have to diffuse from these red blood cells up to the epithelium because it is not vascularized. Okay, we have to form a nice impermeable barrier, and so we can't have anything going through these cells, so it's not vascularized. This would be the apical space. So you can tell that it's a single layer because you can see each individual nucleus very clearly in each cell. The nuclei are somewhat elongated or oval-shaped and located closer towards the basement membrane. And so this is simple columnar epithelium. Simple columnar epithelium. Now, a lot of times when you see columnar epithelium, it's going to have some sort of apical specialization. And here, you can see a darker line on the apical surface. That darker kind of short fuzzy line, that's actually microvilli. And remember, microvilli are actual invaginations of the plasma membrane, which increases the surface area of the plasma membrane of these cells. And an increased surface area means more absorption. So where do you find simple columnar epithelium with microvilli? You find it in areas where you have lots and lots of absorption taking place. Ideal location, digestive tract, stomach, small intestine for sure, because the bulk of absorption takes place in the small intestine, large intestine. Okay? These are all places where you find simple columnar epithelium, where you need lots of, with microvilli, because, well, you have a taller cell, which means more plasma membrane. Taller the cell, more plasma membrane. But you also have this short, stubby microvilli, which means invaginations of that plasma membrane, increased surface area, increased absorption. Okay? So if I have the pointer here, you would say the specific would be simple columnar, and the general would be epithelium. Here's another image of simple columnar epithelium. This is from the intestine, most likely the large intestine. And I just wanted to point out, so again, this would be the basement membrane. This would be the apical space, right? So basement membrane, because this is all connective tissue. You can see the nuclei are, oval shaped, located towards the basement membrane, but it's not really super jumbled. It's easy to tell that this is a single layer of cells, and look the height of these cells. They're columnar. So simple columnar epithelium is just smushed in between the simple columnar epithelial cells. You see these white kind of blobs. Sometimes they may be stained a little pink. These are goblet cells, which produce a lot of mucus. And so a lot of times when you're looking at simple columnar epithelium, you will see goblet cells smushed in between them. And goblet cells, the reason why they're white is they produce a mucus and a lot of lipid. So mucus means high lipid content. And a lot of times on histology slides, lipids actually get extracted through the process. And so it's white. It's actually absent of whatever was contained in those cells. So it's a white or clear appearance. So these are mucus-producing cells called goblet cells, smushed in between the simple columnar epithelium. Okay. So again, if I have the pointer here, you would tell me simple columnar for specific, epithelium for general. And these all are goblet cells. Now, remember I said goblet cells because they are shaped like a goblet, that you can see them as clear or pink. You never want to learn slides by color because there are hundreds of histological stains. So you never ever want to learn by color. You will always want to learn by structure. So it doesn't matter what color stained slide I give you, right? You should be able to notice that these cells are there's a basement membrane, there's an apical space. So if I have the pointer along here, I'm asking an epithelial type, right? Free apical space, basement membrane. Then you should notice that it's a single layer, so simple. These cells are taller than they are wide, so they're columnar. So they're a simple columnar epithelium. This slide could be silver stained, it could be purple, it could be blue. You have to know structure, not color, okay? Identify these tissues based on their structure. Here's another super beautiful, simple columnar epithelium, right? So this would be where the basement membrane is because it has all this connective tissue below. Up here, this would be free apical space, right? Again, look, you can tell these nuclei. This is a single layer of cells. Nuclei are somewhat oval-shaped, elongated, closer towards the basement membrane. And these cells are taller than they are wide. So this is simple columnar epithelium. And this is actually of the stomach. Okay, so simple columnar epithelium. And all this fuzzy stuff towards the apical surface, that's all microvilli. Okay, so anywhere where you need lots of absorption taking place because you increase the surface area, increase the amount of absorption that can happen there. Okay, more plasma membrane, more absorption. This is pseudostratified columnar ciliated epithelium. And yes, it's a mouthful, but you have to tell me all of it. Pseudostratified columnar ciliated epithelium would be general. So again, if I have the pointer along here, look, you can tell, first thing you should be able to see is that this is an epithelium because there's a basement membrane with connective tissue below, and more importantly, or more easy to identify, is the fact that there's a free apical space that's open to the outside. So this is a epithelial type. Well, if you're looking, you should immediately think columnar because these cells do look taller than they are wide. But the nuclei, those dark little circles, are very, very jumbled. They're not nice and neat, like when you look at this simple columnar, or even this simple columnar, or this simple columnar. Okay, nice and neat nuclei, for the most part. Okay. The nuclei are very, very jumbled, and so it gives a stratified-like appearance, but it's actually a single layer of cells. So that's why it's pseudostratified columnar ciliated epithelium. Dead giveaway is if you see the cilia, okay? The cilia. So on the free apical space, you see these really long finger-like projections. Those are all cilia, and they're much longer. If you compare, look, you can barely even see, this is just a fuzzy line on the apical surface. That's microvilli. They're very short and stubby. Microvilli, very short and stubby. Microvilli, very short and stubby. Go here and look, these are much longer if you compare the two, right? So cilia are much, much longer, finger-like projections. So dead giveaway that this is pseudostratified columnar ciliated epithelium. These guys, again, these are goblet cells that produce mucus, and that mucus would go to the free apical space. Okay, it would be secreted out here. So where do you find pseudostratified columnar ciliated epithelium? Ideal location to tell me is in the respiratory tract, so in the trachea, your wind, quote-unquote, windpipe, your bronchial tree, your bronchi. Okay? Those are the tubes that start to lead to the lung tissue. So why do you find pseudostratified columnar ciliated epithelium in the respiratory tract, like the trachea? Well, having these goblet cells producing mucus to the outside, that mucus, high lipid content, very viscous, very sticky. That means dust, debris, microorganisms, things that we do not want to get down to the delicate lung tissue will get trapped in this mucus that's lining the apical surface, okay? So goblet cells produce a mucus that traps dust, debris, microorganisms. And the cilia, what's the function of cilia? Movement, motility. So these cilia would actually beat upwards, okay, up towards the oral cavity so that when dust, debris, and microorganisms get caught or trapped in that mucus lining the apical surface, the cilia beat upwards and push that junk out so that you can cough it out, okay? So that it doesn't get down into that delicate lung tissue. And if you remember, the lung tissue, lungs are made up of simple squamous epithelium, a single layer of flattened cells. So we don't want dust and debris and microorganisms to get down to this single layer of flattened cells because it would be really easy for microorganisms to cross. Okay, or that dust and debris could clog and make diffusion of gases very, very difficult, right? So, pseudostratified columnar ciliated epithelium is to the goblet cells trap, produce the mucus to trap, and then the cilia for motility beat up and out, okay, to get rid of dust, debris, and microorganisms. Okay. You also find some pseudostratified columnar ciliated epithelium, and it's stereociliated, but we're not going to go into those differences just yet. You find it in like the oviducts, so the uterine tubes. Okay, you can also find pseudostratified columnar ciliated. I know it's stereociliated, but for now you just need to know ciliated epithelium there as well. And think of or it's ciliated. The oviducts are ciliated, so to move the egg towards the uterine body. Yeah. Think it's the epididymis of the male reproductive tract that has pseudostratified stereociliated epithelium. Yes, I'm sorry about that. So let me clarify. You find pseudostratified columnar ciliated epithelium in the oviducts of the female reproductive tract to help beat and move the egg towards the uterine body. And then in the epididymis of males, you have pseudostratified columnar stereociliated epithelium, but you don't have to worry about what stereociliated means. But easiest thing to say is the respiratory tract, right? The trachea. Here's another super beautiful image of pseudostratified columnar ciliated epithelium. You can see it has definitely a columnar appearance. They're very tall cells, apical surface. This apical specialization, look at the really long finger-like projections. Those are cilia. Down here would be the basement membrane, and this would all be connective tissue below. So if I have the pointer here, you would tell me pseudostratified, because if you look, not every single cell is touching the apical surface, but every single cell touches the basement membrane. So it gives a very jumbled appearance. So pointer here, pseudostratified columnar ciliated would be the specific, epithelium would be the general. Dead giveaway, look for cilia. If you see those super long finger-like projections, you're going to tell me pseudostratified columnar ciliated epithelium. If you do not see those long finger-like projections, but you just see a short, stubby, kind of fuzzy border on the apical surface, this is simple columnar epithelium with microvilli. Another very beautiful image, again, this would be basement membrane, all the connective tissue below. It's actually a single layer, but it has a very jumbled appearance, so it looks as if it's stratified. And all those super long finger-like projections, this is all cilia. So pseudostratified, columnar, ciliated, specific, epithelium, general. And last of the epithelial types is transitional epithelium. So if I have the pointer anywhere along here, you're going to first know that it's an epithelium type. Why? Because there's a basement membrane and connective tissue below, but really there's this free apical space. So it's got to be a lining. So definitely epithelium. Now looking through this area right here, you can tell very clearly that it is a multiple layers. So here's a cell, there's a cell. These are multiple, there's multiple layers of cells here, right? But remember when we look at stratified squamous epithelium, which is a stratified, it has multiple layers. As we move up towards this free apical space, look, these cells get much more squished and flattened. When we look at transitional, as we move up towards the free apical space, look how big and fluffy and round these cells get. These are actually called balloon cells, or not balloon, they look like little balloons. They're called dome cells, right? And so as we move up towards the free apical space, they get fluffier and more round, right? Look how round and fluffy and balloon-shaped these cells are. So this is not stratified squamous, right? This is transitional. As we move up through the layers, it looks as if they change shape. Transitional, okay? Where do you find transitional epithelium? Think the urinary system. So you find transitional epithelium in the ureters, which lead urine from the kidneys to the bladder. The bladder, if you get a bladder slide, you will see transitional. So the bladder on the inside is lined with transitional epithelium. So this free apical space would actually be filled with urine in a full bladder, okay? So this, and in the urethra, which it conveys urine from the bladder to the outside. So in the urinary system, you find transitional epithelium. Get a bladder slide, you will for sure see transitional. So why do you find transitional in, say, the bladder? Well, you can imagine that if the bladder, so what does the bladder do? It fills with urine, and then it contracts and it releases urine, and then it fills with urine, and then it releases urine. And so it's constantly filling up, releasing, filling up, releasing. Well, as the bladder fills up with urine, if the bladder was lined with a single layer of cells, well, you wouldn't be able to stretch it very far, right? There would be a very limited capacity to the bladder, to how much urine it could contain. So having these multiple layers of cells means that as the bladder fills with urine, these cells can slip past one another and actually expand larger and contain a larger volume, a higher volume of urine. So it has the ability to, these cells slide past one another so that it can expand. And having these super fluffy dome cells, right, on the apical surface means that as the bladder fills with urine, these can also be flattened down, okay? So this is transitional epithelium. So again, I like to show a couple of images because you can see that they all look a little different to an untrained eye, right? And they may have different colors or different stains, but the structure is totally the same, and that's what you have to learn is the structure, and relate the structure to its function. So this would be the basement membrane, all the connective tissue below. This would be the free apical space where the urine would be. You can tell that as we move up, it changes shape. So this is transitional, but it's definitely multiple layers of cells. These are not columnar, right? And these really super fluffy balloon-shaped cells, those are dome cells towards the apical surface. So these cells can slide past one another to expand and be flattened as the bladder fills with urine. All of this junk right here that's on the top of the cells, this is actually a lot of mucus. So these cells have the ability to produce mucus to help protect themselves because the urine can be very caustic, or it can be, because it's all the waste that we're getting rid of, right? So it can be caustic and harmful. So a lot of times you'll see this big, thick mucus layer, and that's just to help protect the cells of this transitional epithelium from the toxic urine, okay, or things that could be toxic to the cells. So if I have the pointer here, you would say transitional would be the specific, epithelium would be the general. Okay?